explore the biggest questions of our time, and get to know the people behind the science. This is Curiosity Meets Discovery, research talks from inside the Max Planck Society.
Despite covering over 70% of our planet, the depths of the ocean still hold many surprising secrets. Not only have significant portions of the ocean not been explored, but there are many species of living creatures that we still don't know about or understand. For example, did you know that there are creatures living in the ocean that have no mouths and no guts? And some of them thrive in environments miles below the surface, near hydrothermal vents that were once thought to be incapable of sustaining life. How is this possible, and what can we learn from it? Well, today's speaker, Dr. Nicole Dubilier, is diving into these mysteries, literally. And her research program is focused on a very important biological phenomenon that often gets overlooked in thinking about marine life: symbiosis, a mutually beneficial relationship between animals and bacteria that makes it possible for both to thrive in environments that neither could survive in alone. But exactly what is symbiosis? How does it solve the challenges of life for the species involved? And what insights can we glean about our own body's relationships with bacteria? Dr. Nicole Dubilier is a director at the Max Planck Institute for Marine Microbiology in Bremen, Germany, where she heads the Symbiosis department. She's also a professor at the University of Bremen. Dr. Dubillier's work has had a significant impact on our collective understanding of the importance, diversity, and function of symbioses between bacteria and animal. Her research has provided a critical contribution to marine microbiology and ecology, and I am excited to welcome her here today. Nicole, welcome to Curiosity Meets Discovery.
Thank you for inviting me to give this presentation. I'm excited about the opportunity to present my research. The title of my talk is Living Together in the Dark: Symbioses between Bacteria and Marine Animals. And why we call it Living Together in the Dark and what symbioses are, I'll explain as we go along. What you see here are pictures of mussels that are full of bacteria that live in symbiosis with them at hydrothermal vents, and I'll describe how we discovered them and why you might actually find that that's relevant to the microbes in your own body. So in addition to the mussels that you just saw, one of our favorite lab rats, which means the animals that we like to work on, are very, very small, minute, gutless marine worms. And what you are seeing here is a greatly magnified image of this worm. And in reality, they're barely visible with the naked eye, so about an inch and maybe long and a tenth of an inch in diameter. And so you might ask yourself, well, why in the world would anybody want to work on, this little worm without a mouth or a gut, and why should we care? And I hope that I can make it clear why we care. It's actually fascinating because this worm is full of bacteria that are so efficient at providing it with food and nutrition and recycling its waste compounds that it has lost its mouth and gut, its digestive system throughout evolution. The closest ancestors have a gut, and they took up these bacteria and they became gutless. In all honesty, there's another reason I like to work on these worms. The main distribution of these worms is in tropical environments. We find them particularly on some of the most beautiful islands. We get to sample that you see here in part Belize and Bermuda, where we've sampled them. And actually, one place that I've never gotten to, but I'm dying to sample, is the coast along Florida, both in the Atlantic and in the Gulf of Mexico, where there are reports of these worms, but where I've yet had the chance to go to collect them myself. So what is symbiosis? How does it work? How do these associations work? So symbiosis was defined quite a while ago in 1879 by a German botanist, and he was working on lichen, which are the things that grows on trees, and he was looking at them more carefully, and he discovered that these lichen are actually the living together of an algae and a fungus. Fungi are mushrooms. And so we defined the term symbiosis as the close and long-term living together of unlike organisms. And I like to emphasize when I teach, we're not talking about a husband and wife living together. It's essential that it's two different species. So Homo sapiens among each other may have, psychologically speaking, symbiotic relationships, but in biology, we are talking about different species coming together. And what we, in terminology, we always call the host the larger partner and the symbiont the smaller partner. And I'll be using these terms regularly from now on. What I'd like to emphasize is that for literally centuries, it was assumed that the diversity of life that we see on Earth is driven by competition and predation. But this view is one-sided, and it completely fails to capture one of the most fundamental drivers of evolution in life, and that is symbiotic associations, intimate co-evolved interactions, beneficial interactions between species. We've been looking at symbioses on land for quite a while, for centuries, and because simply we're land animals ourselves, and it's much easier for biologists to work on land. But the oceans have the highest biodiversity, and 70% of our planet's surface are covered by the oceans, but they're harder to get to. And what I'm going to tell you is how we discovered these gutless marine worms that live in shallow waters by diving down deep with a submersible, I'll come to it, from the Woods Hole Oceanographic Institute, and discovering hydrothermal vents, and then realizing, oh my God, this is, these kind of worms are everywhere. So in the early 70s, geologists were looking for underwater hot springs. They had realized for about 20 years that the Earth's plates are spreading apart, and they knew that in the oceans where the Earth's plates are spreading apart, that there should be hot water springs, underwater hydrothermal vent springs, very similar to what we know when we see them above on land, like in Iceland. And so there was a bunch of geologists. They got together on a cruise here with the Woods Hole Oceanographic Institute. We call it WHOI, the WHOI Institute in Woods Hole, on their ship Atlantis. And it was also a period of discovery in the oceans, because submersibles were still very, very rare that any research institute would have them. They were mainly used by the military for military purposes, including looking for submarines and all sorts of other purposes. And so there were indications that off the Galapagos Islands, there were temperature increases in the deep water, and that hydrothermal vents might be a place to look for off the Galapagos Islands. And as I explained, the geologists were fully aware, and this holds true too today, that through volcanic and tectonic activity, the Earth's plates are moving apart, and they form these underwater mountains or ridges where fresh magma comes up and is growing, and where we see both the continents drifting apart, and in other zones we see subduction zones like we know from California. Now, most of the deep sea is a desert. So when you go down with a submersible, 4,000 meters water depth, these are images of what it looks like. And these are exciting images of what the deep sea looks like, because you actually see more than just mud. And so we see a sea star feather here on the right, and a sea cucumber on the left. But that's pretty much it. And otherwise, there's a brittle star here on the bottom of the one image. You're going to see tons of mud for miles and miles and miles. And the reason that we see so little life is because there's so little food in the deep sea. And the reason there's so little food in the deep sea is because it was assumed previous to the discovery of hydrothermal vents that the only way large animal communities can get food is through photosynthesis. The primary production, just like plants on land, in oceans, we have little tiny algae, and they're using photosynthesis to fix CO2 into organic carbon. And a lot of that is being done at the surface, but very little of that food that is being produced makes it to the bottom, because there are all sorts of little animals in the oceans that are already eating it up. And we estimate of the 100% of, let's say, of primary production that is occurring in the oceans, only about 5% makes it down to the deep sea. So you can't actually fuel large animal communities in the dark. That was the assumption, that the deep sea is a desert. And so what I'm showing you is similar to what these geologists that went down with Alvin in the submersible saw. They're looking for the hydrothermal vents. It's kind of dark and they're seeing lots of mud. And you have to remember they're very, it's very dark. There's only like this one spotlight or two. And bingo, they go, oh my God, look at this. There are these gigantic animal communities down here in 3,000 meters water depth. And they had never seen animal communities like this. And I think these are beautiful tube worms. You can see they've got, a giant red plume on the top, and they live in these tubes. You can also see that there are mussels that occur here. And here we're zooming in. And it's reported that the two geologists that were in the submersible called up to the mothership to Atlantis, and there was a student that had studied a little bit of biology, and they on the call up, they said, isn't the deep sea supposed to be a desert? Like there aren't supposed to be any animals down here? And she said, yep. And they said, well, there are a lot of animals down here. And it's reported that they grabbed a few of these animals, and because they aren't biologists, they didn't know how to fix them. And it was back in the days when research cruises weren't dry US research cruises, so they had a lot of vodka and whiskey, and they fixed them in that and brought them home to the labs in the US that looked at these giant tube worms. They're as thick as my arm and big and long, and discovered that they didn't have a mouth or a gut, and that they were full of symbiotic bacteria. And I'll explain how that symbiosis works. So the big question, of course, was where is this food coming from at the hydrothermal vents? What are all these animals living off of? And the way the food chain functions at these hydrothermal vents is the hot fluids that are coming out from being heated by the magma in the deep earth. These fluids contain chemical compounds such as hydrogen sulfide and methane, and bacteria can use these as energy sources to fix CO2. And we call this way of primary production, in parallel to photosynthesis, we call it chemosynthesis. And I'll explain it in a second. And it's chemo because it's chemical compounds that are used to fix CO2. Photo is from light. So it became clear with the discovery of the hydrothermal vents that you could indeed fuel such large animal communities through chemosynthesis. So I'm going to do a little bit of your basic biology 101. Most or many people are familiar with photosynthesis. It's the energy that comes from the sun. It allows chloroplasts, which, by the way, are also symbionts, descendants of cyanobacteria that colonized ancestors of plants a long time ago. And the chloroplasts use the energy from the sun to fix CO2 into organic compounds. And that they're building the biomass, they're passing that on so the plant can grow. And in chemosynthesis, what we have is the chemical energy that is provided by oxidizing reduced compounds like hydrogen sulfide and methane is used by bacteria to fix CO2 into biomass, into sugars and carbohydrates, and they then feed their host. Hydrothermal vents are called the rainforests of the deep sea. The biomass is impressive. It's as high per square meter as it is in rainforests or in coral reef environments. And what excited the biologists about discovering them is that the entire basis of these communities are symbioses between the animals and the chemosynthetic bacteria. Animals can't do primary production. They can't do photosynthesis and they can't do chemosynthesis. They can't use the energy at vents such as hydrogen sulfide and methane to fix CO2 themselves. So what do? They're down there and they say, oh, well, here are some partners that can use the energy sources that are here. And over evolution, they hooked up and came together with them to be able to thrive at these nutrient-poor sites. And in retrospect, what still continues to amaze me is that these types of symbioses are actually in our own backyards, and I'll come to that. As I just described, we discovered them in the deep sea at hydrothermal vents, and then biologists said, oh, well, cold seeps usually where oil is found. We know that we often find shells of mussels and clams that you see at vents, and they very quickly realized that this is another site in the deep sea where we can find these symbioses and whale falls, which are very, very cool phenomena where whale falls sink to the bottom of the oceans, the meat on them, the dead whale falls to the bottom of the ocean and the meat is quickly eaten by sharks. But then their bones begin to decay and produce a lot of hydrogen sulfide, and we also find these symbioses. But in retrospect, what is amazing is these symbioses are in what we call our own backyards. You can walk into shallow waters off the coast of Florida, and you can find animals that have these types of symbioses in the sediments, in seagrass environments, in coral reef sediments. And biologists for the longest time had wondered why there were animals in shallow water environments that didn't have a mouth or a gut and how they were living. And they assumed maybe they can take up nutrients through their skin. But going all the way to the deep sea to understand the symbioses enabled us to understand what we also have in shallow water environments. And what we now know after working on these symbioses for over 40 years is that these types of associations have evolved multiple times. This wasn't just a one-off affair that happened once in the deep sea and then colonized shallow waters. Not only do we know that they occur everywhere, most likely they're still happening. There are still animals coming together with bacteria to create new symbioses. So we talk about them having evolved multiple times independently of each other. And what you see here is a tree showing the relationships between the different types of bacteria that are associated with different types of hosts that are drawn at the branch ends, so worms and clams and all sorts of animals. And so we know that at least 15 different types of bacteria have established these symbioses. And we also know that two single cell groups of eukaryotic organisms called protists and 9 animal groups have established these associations, snails and clams and mussels and worms. And what that tells us as biologists is that there's obviously a strong advantage for both the bacteria and for the animal hosts in coming together. When we work in the deep sea, it's always a challenge. What you see here are the research vessels with which I've done a lot of my research in Germany, the German research vessel, the Meteor, and we work with autonomous underwater vehicles here that are actually flown by pilots from the ship. And cruises like these are very expensive. We estimate that they cost something like $50,000 per day. And if you're unlucky and the weather is bad, then you can be out at sea for weeks and not even get your submersible into the water. So this is a challenge. And so working with shallow water animals can be a lot easier. And this is the sophisticated equipment we need to collect the little tiny worms that I showed in my first slide. You need a bucket and you need a sieve and protection against the sun. And the two gentlemen that I'm showing on these slides are the founding fathers of the research on the gutless oligochaetes, Olaf Dürer, with whom I did my PhD, and Chris Arzeos, who is the only person in the world who can go out into the field and actually identify these different worms. So when he retires, I have a problem, although molecular tools are allowing us to identify these worms without looking at them morphologically, looking at their bodies. So now I'm going to show you a little video of what it looks like when we collect these worms. It's a little bit more sophisticated because we go diving for them. We've been doing a lot of work in the Mediterranean in an Italian island called Elba off the coast of Italy. And these orange buckets that you see are for collecting a lot of sand, because we look for these worms, but we have to sieve them out of the sand. And we simply, by going to certain sites for many years, start to know what are good sites to collect them and what are bad sites, like people that go out clamming and know, yeah, this is always a site where I find a lot. And what you're going to see now is that the diver is going to shake up and stir up the sand. And what happens then is any animals that are lighter than the sand are going to swim up into the water. And you're going to see these little white threads. And the diver is going to show them against the neoprene suit that they have, their wetsuit. And you see that little white coiled thing. That is one of our worms. You can see I can get still very excited about these worms. And so what we do is that helps us identify a site where we know they're going to be. And then we start collecting sand, we call marine sands or sediments. We start collecting them from this site and then start sieving them intensively in the lab. One of the important tools we use is to identify what are the energy sources that the symbioses are using. What you see here is we're sticking long tubes into the sediment, and we're pulling out the water, the pore water that is in the sediment or sand, to better see what kind of energy sources could they be using. Do they have the hydrogen sulfide that we think their symbionts might be using, or could they be using other energy sources? Do they have enough oxygen? That's important. They need it to breathe. And what you see here is then these incredibly heavy buckets of sand and sediment that are full of our worms that we're taking back to the lab. And when we get back to the lab, we have quite a sophisticated toolbox that we use to look at the symbioses. I described some of our experiments that we do, or the work we do in the field, also with experimental work where we for example, in the deep sea, displace our mussels from venting and they lose their symbionts and can regain them. We use a lot of imaging tools, as biologists do, to look into the symbioses in more detail. We do modeling to try to understand how the symbionts pass on their nutrients and the host takes them up. And then we use molecular tools, genomic sequencing, transcriptomic sequencing to look at the genes that are present in both the host and symbionts and which ones are being expressed in the symbiosis. And so this little gutless worm, it harbors its symbionts directly underneath its skin. And what you are seeing here on is a cross section through the worm. So we take these super sharp knives, we section them, and we look at them under a microscope that is called a fluorescence microscope, and use labels for the symbionts. And what you see here, fluorescing in green and in red, are the different types of symbionts that are present underneath the skin of this worm. So I like to compare it to this giant like bicycle tire, where directly underneath the rubber, you've got all these bacteria sitting in there. It's held together by a muscle layer of the worm, and inside there's actually just fluid and a little bit of organs that I won't bore you with. And so this entire worm can get its entire nutrition and recycle its waste compounds from this inner tube of bacteria that are providing it with everything it needs to live. And the way the symbiosis works is the worms eat with their skin. So now we're zooming in a little bit more in more detail into the section that you just saw directly, the inner tube section. And normally all animals have an outer layer of their skin that's called the cuticle, and they've got epidermal cells that sit right underneath it. And what has happened here is that this thick layer has pushed down the epidermal cells, and there's a layer in which they're sitting. And the amazing thing is, because these worms no longer have a gut, what we're seeing is they're using enzymes that they're normally worms would use in their gut to digest the food in their gut. They're using that to eat up these bacteria. And the way they do that is they digest them in what are called the garbage cans of cellular products, these lysosomes, which normally eat up, let's say, cells, organelles and cells that don't function so well, like mitochondria or something like that. And that's like a waste bin. And here the bacteria are constantly being eaten by these lysosomes. I like to, I'll start wrapping up, and I'll like to expand in the hopes of that beyond just saying, oh, well, that's kind of really cool, that there might be other reasons why you might actually, there might be value in looking at these types of associations. And there was a joke that appeared in the New Yorker quite a while ago when the hydrothermal vents were first came out, and everybody was excited about talking about them and what was going on, but it was also hard to understand. And so this cartoon really rings true to me that it's not always easy to understand biologists' excitement about what they're working on. When I started working on these symbioses in the field of symbiosis over 30 years ago now, it was assumed that most of the microbes that we as humans are associated with are actually pathogens that we need to actually constantly figure out how we can fight against them, use antimicrobial compounds, antibiotics to get rid of them. This was the vast assumption. And what has completely changed in our understanding of our interactions as humans with microbes is that the vast majority of the bacteria that are on our bodies and inside our bodies, inside our guts, are actually beneficial. And those of you that have taken prebiotics and probiotics, that have thought about what antibiotics do to your gut microbial communities, know how important our microbes are for our health and well-being. And because these interactions are based on a certain set of genes and interactions that are limited, some of the work we're doing on our very, very simple organisms is relevant to the extremely intricate interactions that we as humans have with our microbial communities. So I'll finish up by saying why I think, not only I, actually a large now proportion of biologists, evolutionary biologists, have realized the importance of symbiosis. So the remarkable diversity of life that we have on our planet did not evolve solely through competition. All plants, all animals, including humans, do not live alone. We live in close intimacy with millions and millions of beneficial microorganisms that are essential for our health and well-being. And cooperation and symbiosis played and play a key role in the evolution of life. And without symbiosis, life on our planet, as we know it today, would not exist. And with that, it's been a wonderful, or is a wonderful symbiosis also with the people in my group that have been doing the work, some of which I was able to show you, and many of the funding sources that have allowed us to do our research. And thank you for your attention.
Thank you for that truly amazing talk. I mean, the first, I couldn't help myself when I'm watching it. It's a beautiful way to demonstrate curiosity-driven discovery in the sense of who would have thought going to the bottom of the ocean would have taught us about, you know, this new chemosymbiosis. I mean, that's I think it's absolutely fantastic, and it's important for people to realize that we never know what we're going to find, and we never know exactly how that's going to change what we thought we knew.
I couldn't agree more. I think it's very hard for non-scientists to understand why would anybody want to work on, you know, this little worm, like, and what does that tell us? But there are two aspects. The one is funding researchers just to be curious and to say what's out there, and then to take it to that next step and to say, well, what does that tell us and why should I care? I think that this is so essential for basic research.
You know, it's really cool and interesting to think of how this relationship gets set up. You know, what is it that, you know, allows the bacteria to find their host? And also, for that matter, what is it about the host that recognize, hey, you're a good guy, hey, no, elsewhere. And I'm sure that a lot of effort has gone into trying to understand the mechanisms that might be responsible, the signaling mechanisms that might be responsible for that.
Yeah, in fact, I was convinced, David, that at working as a neurobiologist, neuroscientist, that you would ask me, you know, do they have any nerves in their bodies, and in what we call a brain-like organ in these worms? Do they use that to navigate possibly towards environments that are attractive for their bacteria or away from ones that are unattractive?
So you put this in the context of evolution, which I think is really interesting. But, you know, I have a hard time understanding, you know, how could symbiosis actually evolve? I mean, in the end, you can say, oh, it's beneficial for both parties, therefore it works. But the initial stages of a symbiotic relationship must be, how do I say it? There must be many years before that evolves into something that has, you know, the benefits for both. Do you have any insights into that? I mean, maybe development would give us some insights into that.
You're in good company in that that's a question as symbiosis researchers we are constantly asking ourselves, which is what we're looking at is the endpoint of a perfect symbiotic association, but the first steps, the coming together in evolution, that is one that we don't have a record of. So what we need to do is look at, for example, hosts that have symbionts, and then closely related hosts that don't have symbionts, and then possibly expose them to the symbionts and see what happens. There are some things we know now, which is bacteria love to colonize any surface. And they particularly like to colonize in the marine realm. They, anybody who's had a ship knows what the problem is, or a boat, right, with biofouling. Yeah. And put a glass slide into the water, it's immediately colonized. And so you're creating these communities. And animals leak out waste compounds all the time. In fact, that's why our skin is so colonized. We're constantly excreting stuff. And that's valuable food for bacteria. So bacteria, one of the first steps is bacteria are attracted to animals and plants because they're excreting waste products that they don't use. And the bacteria are always microorganisms, archaea, viruses, they're all looking, to attach and get some of that food that is being excreted. Now, animals, and particularly those that are in marine realms, they don't like to be overgrown, right? They don't want to become all overgrown because then they can't breathe. They need oxygen to breathe. So you could envision that there's this dance between the bacteria saying, well, hey, I'll be really nice to you. I'll eat your waste compounds, but I won't overgrow you too much. And what that implies is that there has to be communication between the two partners. They have to start to be able to recognize each other. They have to start to be able to communicate. It's a very intricate dance, and thinking about it is exciting. We can only guess at the actual steps so far for our symbiosis.
Right. Yeah, I was also thinking of it from the developmental perspective. And I'm guessing, I guess one of the other big words from your talk today is diversity, right? The amazing diversity that nature has in terms of relationships between the bacteria and the host and so on and so forth. But I was wondering, an animal that has no gut, does it start out? When it's born, does it have no gut? Or is that something that emerges as it develops? How in the world did that get selected for?
There too, the diversity that nature has come up with doing that is absolutely amazing. We're just beginning to work on it, but I'll tell you how it is in our teeny tiny worms, and then I'll tell you what we know about the deep sea worms. And the pathways that evolution has taken to get rid of your gut are completely different. So this is not
two different worms.
Completely different. The worms aren't related.
Okay.
So in our gutless worms, in our teeny tiny small ones, the ones that occur in shallow waters, all evidence indicates they no longer, even in an embryo in the development, they don't develop a gut. They don't even bother anymore. There's nothing there that we can find. We're now very carefully looking at the genes that are expressed during the developmental stages, because we want to know, well, how do, for example, the enzymes that I talked about that are expressed in the skin, are they then expressed throughout the entire development, or how do they, normally they should be heading towards the gut and now they're in the skin, how that works? That's what we're still looking at. Now, those giant tube worms that I was talking about and showed the video of, that's been looked at by colleagues in the US, but also by a colleague in Austria. And she's done some beautiful work. And there, it's the most amazing thing. I love this story. What happens is they have when they reproduce, they build something called a larvae. This is what a lot of marine animals do, mussels, clams, and so on. It's a dispersal phase. It's a very, very early developmental stage of many marine animals. And this larval stage has a mouth and a gut, looks like a perfectly normal, developmental larvae. And so it was assumed that these larvae that are eating also the plankton, that they would, they've got a head. I'm now going to pretend I'm a larvae and I've got like my mouth to feed on and I've got my butt back here. And it was assumed that they would just kind of like, close off their mouth and close off their butt. And that would then become this huge organ that they have in which they house their bacteria. What happens is they close off their mouth and their gut, but their symbiotic bacteria infect them through the skin and create a whole nother organ in which they live from another mesenterial, it doesn't matter, but another tissue. So talk about diversity. And there are many, many ways to skin a cat, or many different ways that both animals and microbes have found to adapt to each other. This is, for me, a beautiful example of that kind of diversity.
Oh, no, amazing. Has there been any thought to climate change, increasing the temperature of the waters and so on and so forth, to how that might impact these symbiotic organisms? I mean, it seems to me that how do I say it? A lot of things can go wrong in a symbiotic relationship, right? And is there any evidence for that sort of thing happening? And I'm wondering whether our ecosystems, this would be another aspect of our ecosystems that climate change could impact and that we should be paying attention to.
So we don't see the kind of dramatic effects we know from coral reefs, where we see the bleaching events that are really so disturbing and really painful to watch. We can only make assumptions so far because they're buried in the sediment. Let's talk first about the shallow water hosts. They're buried in the sediment. The temperature differences there are better buffered than in the water. Anybody who goes swimming in the ocean knows how it's nice and warm in the surface. And then as you go deeper, it gets colder. So in the sediment, it stays pretty constant. It will be interesting to watch in the Mediterranean. It's heating up very quickly, and it's also affecting the sediment. So if there are effects, we might, it's depressing to think that we might be able to research them, but the next 20 years will tell if even in these more temperature stable environments, we can see an effect. In the deep sea hydrothermal vents, what we're worried about is deep sea mining. That's a issue. If we want to use, already our computers, if we want to use electric cars, we need rare minerals, and it's very expensive to do that on land. And hydrothermal vents are full of a lot of valuable metals for technology. And there have been companies that have tried to raise the money to do deep sea mining. Luckily for biologists, I would say, and for anybody who cares about the ocean, so far it's been too expensive and it just hasn't been valuable. But when you think about the effects of what mining does on land, you can imagine what it will look like also in the oceans. And so climate change, yes, that might eventually affect the deep sea, but estimates are it's going to take a long time for the actual deep sea to heat up. And these animals are close to hotter temperatures. But if it gets too hot, they're not going to like it. But by that time, I don't know if humans could live on Earth if it gets that hot.
Yeah, no, I hear you. So my last question for you is kind of out there, but I wonder. I mean, you can really sense your passion for what you do. I mean, it's very clear in how you talk about it and all that you've achieved. Is there something that you wish you could do in terms of the science? Is there something that, if I could only do X, that you feel like it would have, a huge impact on our understanding of these symbiotic relationships?
Yes. Actually, the bucket list is very long, but I'll try to be concise. To this day, we have not been able, no one has been able to grow the symbionts in isolation from their host. So we can't, yeah, exactly. You would think it shouldn't be so hard. Yes, exactly. And we haven't been able to do that. And in fact, when I started out in this field, it was almost one of the biggest reasons my proposals were rejected, which is, hey, if you can't cultivate the bacteria, if you can't genetically manipulate them, why in the world would you want to go to the bottom of the sea and work on these exotic panda bears of the deep sea? Yeah, that's what I used to call it. Everybody likes panda bears, right? But are they important for ecology? Do we care? And we can't cultivate them so well either. Well, we're getting better at it. Anyway, sorry, that was a little excursion. That would be on my bucket list, that I would be able to cultivate them and test. Then I could examine more closely how they colonize their host, how they can persist in them, the communication between them, because I would look at hosts with and without the symbionts. I couldn't do that in the gutless worms, but there are some that still have a gut. We have these intermediate stages. So yeah, that's big on my wish list. When PhD students join my lab, I tell them that they're also very gung-ho to give it a try. And then I say, all right, you've got 3 months, because if it doesn't work, you don't have a PhD. So that's the deal.
Yeah, so give it all you have for 3 months, and then yeah, no, I hear you, but that would be huge. I mean, to watch that in front of you, using their fluorescent tags, et cetera, to actually see it in action, that would, yeah, exactly. I hope you get that, okay?
Okay, I hope so too.
All right, Nicole, thank you so much for this presentation. Really enjoyed it.
Right, and I enjoyed it too, David. I'm looking forward to ringing you up someday from, who knows, from Sanibel Island and going, I found them, I finally found them in Florida.
My apologies. When you said that during your talk, I was going to say, Jupiter, Florida. Come on down, okay?
All right, okay. All right.
Nicole, take care.
Have a nice day. Bye.
Thanks for watching Curiosity Meets Discovery, research talks from inside the Max Planck Society. Visit mpfi.org to sign up for updates. Follow us on social media at mpfneuro. Curiosity Meets Discovery was produced by the Max Planck Florida Institute for Neuroscience.